So Large PD, which is the Latin American Research Consortium on the Genetics of Parkinson's disease, is as I said, it's an initiative that started in 2006. We had a couple of institutions in Argentina and Peru. And the idea originally was just to, study some of the Mendelian genes in Parkinson's disease and understand if some of the mutations or the pathogenic variants that were found in other populations were also present in these individuals. Obviously, Latinos have a high or at least some percentage of their genome that comes from European populations. So if you see some of these variants in Europe, you would expect to see some of them in Latin America as well. And some of these genes are important because there are ongoing clinical trials and they're fairly common in other parts of the world. So the study started like that. We just wanted to see what it looked like. So we were receiving, a few patients here and there from each of these cohorts. And then, as word got out and we started to do some interesting research and saw that there were differences, between Latin America and Europe and the US, and also even within Latin America between countries, because the ancestry is very different. For example, in Argentina, it's mostly European, while in Peru, it's mostly Native American. So we started to see some differences, and then other groups in other countries started to join Large PD. And in 2016, we managed to build, with funding from the Parkinson's Foundation, we managed to build a cohort large enough to be able to do the first genome-wide association study. So that manuscript that you referred to, which is the copy number, that's the first of two that we have out. So the second one just got published in Annals of Neurology, just came out, I think, a week or two ago, where we present the results from the genome-wide association study, the first GWAS in Latinos with Parkinson's disease. We found some interesting results there, looking at synuclein, which is the top gene for all populations. It's also the top gene in Latinos. So it kind of reinforced the idea of the importance of this gene in Parkinson's disease. Although some of the variants are different, the gene is the same. And then we nominated a few other regions that might be important in Latinos that are not present in Europeans or Asians. And then the copy number variance was, using the same data. We looked at overrepresentation of copy number variants across the genome and also in genes that cause Parkinson's disease. And we found also that a copy number variant in Parkin is a very important cause of Parkinson's disease, early onset Parkinson's disease in Latin America as well. So those are two of the main studies. We have now 36 sites in 13 different countries. And we're also recruiting Latinos here in the US. So we started a cohort of Latinos here in the US. So we're now with COVID, we're doing everything remotely. So we do everything over the phone, and then we send saliva kits to the participants to their house, and then it gets returned to us. And that's how we got the DNA to be able to do the genetic testing. The idea is I think we have about 5,000 individuals recruited so far. And our idea is to double that to almost 10,000 in the next 2 years if COVID allows, with all these new sites. It's a very exciting initiative. And we're also working on families to try to find new genes for Parkinson's disease. So that's one of the next steps that we're working on. So we've run 170, hopefully about 250 total by the end of this year, through a panel that includes all the monogenic genes that cause Parkinson's disease, and then some other dementia genes. And we found that those countries in Latin America that have lowest European ancestry has also the lowest number of pathogenic variants in this gene. So it kind of tells us, and some of these families are like multiplex families where there's more than 5 affected individuals. So there's a clear genetic component, but still the genes that we know of, which come mostly from studying European populations, are not represented in this population. So it kind of tells us that there might be other genes that we are not aware of, that we're not screening for, that might be important. So we have now money to do whole genome sequencing, both through a grant that we have here locally and also the GP2 project that I spoke before. So we'll be doing a lot of whole genome sequencing to try to find new genes that are involved in Parkinson's disease in this population. So.